I am Roberto Toro from the Applied and Theoretical Neuroanatomy Lab at Institut Pasteur, and this is the course on biomechanical modeling of cortical folding. I will start with an introduction. So here you see the cerebrum of many different species of vertebrates. The cerebrum in many of these species is folded, but you have to keep in mind that only mammals have a proper neocortex. So here you see, for example, the first brain on the top left is a shark. Most of what you see is actually not the brain. The brain is very small, right in the middle. Then you have a bird, and then from the rat on, you have different mammals. So the amount of brain folding will correlate with brain size, as you will see. But already, as you may know, the cerebrum of most of the smaller mammals is not folded. Here, for example, for the rat, or in a primate like the mouse lemur. Then you have the brains of kangaroos. So their cerebrums are also sometimes folded. It's important to keep in mind that they are marsupials. So in many cases, they don't have a corpus callosum, for example. Then you have the cerebrums of several different primates, different felids, then a sheep and a pig. You see also the amount of brain folding in different types of canids, like the raccoon dog, hyena, or coyotes. And then also in some bears, like the sloth bear, polar bear, or the grizzly bear. And finally, you see the very nicely folded cerebrum of the rhinoceros. But the cerebrum is not the only, so the cerebral cortex is not the only structure that's folded in a mammal brain, in a vertebrate brain. So here you have, for example, the cerebellum, and in contrary to the neocortex, the cerebellum is folded in many vertebrates. So it would be folded in even some fish and birds. It's quite impressive to see the diversity of the amount of folding in different animals, but it's, I think, important to keep in mind that this is nothing compared with the diversity in what they do and the type of ecological niche they occupy. So some of them can fly, some of them can swim, and they are basically using inhabiting every possible environment on Earth. So why study brain folding in the cerebrum or the cerebellum or in any other place? Well, first, it's interesting to realize that there's a quite strong reproducible relationship between brain folding and cytoarchitecture. Here you see the map that probably most of you know very well, that Brodmann drew based on the differences in layering of the neocortex, or type of cells, the neurons that you could find in different places, etc. And for example, the central sulcus or the calcarine sulcus and many other primary folds are occupying a very prototypical location relative to cytoarchitectonic regions. For example, the central sulcus will divide the somatosensory from the motor cortex. This has not been always clear, so there was a very influential paper by the team of Carl Ziles, where they're suggesting that, as you can see to the left, that soil science borders of architectonic areas coincide only in a few examples, and that in general they are not valid landmarks for microstructural organization of the cortex. And that was quite bad news back then in '97, but luckily a bit more than 10 years later, in a more precise analysis led by Bruce Fisher and with also Carl Ziles as a last author, actually they informed that the relationship seems to be much stronger than what they previously thought. So why study folding? The relationship with the site architecture can also be related to the amount of folding. So to the left, you see an image of the changes in brain folding relative to brain size. And it's interesting to realize that as the brain volume increases and then the amount of brain folding increases, the number of cytoarchitectonic areas, which are not evident to measure all the time, but it seems to increase. That's also interesting. But not only cytoarchitectonic regions, also the amount of connectivity or the presence of connections seems to correspond with the presence of folds. One very strong pattern that it's very easy to see in different species is the U fibers that will connect one fold with the next one. So one gyrus with the next one passing under a sulcus. This is like some sort of a warning. You will see in many of the papers that follow titles that start with on something. And this is all references to a very important seminal work of Darcy Thompson on growth and form. Darcy Thompson, for those who don't know, is like the equivalent of like the closest that would be to Darwin for a physicist interested in biology. So he was interested in the way in which patterns emerge in nature, both in living organisms and also in just non-living matter. So it's a very interesting work. You know already the figures that I'm showing here to the right. Okay, so I will now quickly review some hypotheses and models of brain folding. So there's two main families of hypotheses that I would call the mechanical and the genetic, or gyrogenetic. No, so the mechanical hypothesis, most of the time people would assume that brain folding appears because of the constraint imposed by the skull. That can be tracked back to the work of Legros-Clark, the formation patterns in the cerebral cortex, where he suggested this. But probably there were many other people suggesting this before him. So if that were the case, then folds would appear just because of this constraint of the limited space constraint, and one would expect that they don't have any sort of regularity or eventually even importance. It could suggest that brain folds are epiphenomenal in a sense. However, very soon after, the work of Baron in particular showed quite clearly that actually brain folding is not constrained by the limited space inside the brain, inside the skull. He did some quite dramatic interventions where he removed big chunks of the brain of a sheep in utero, and the remaining brain. So if it were because of the lack of space, one would expect that it wouldn't fold, but it folded with a pattern that was barely the same as in a normal sheep. More recently, Mukli and collaborators showed this case of a person that was born with only one hemisphere. This is very infrequent, as you may imagine. And the remaining brain, as you can see in the MRI images, is folded as one would expect. So if it were because of a problem of space, this hemisphere, one would imagine it would occupy the whole available volume without need to fold. Now, however, that hasn't prevented people from making models about this possibility. There's one old paper by the Raghavan and collaborators where they were simulating here, as you can see, a simple 2D model where the dashed lines represent the constraint of the skull, and then they made a spline grow inside, and here you can see the spline folding, and of course you can imagine that the folds, depending on the properties that you add to your spline, could fit or somehow correspond to the wavelength of folding in real brains. More recently, in Science this time, Bruno Motta and Susanna Herculano-Hussel proposed something that's quite similar. In this case, it's what I would call a crumpling model. So the limited space here was produced by their hands. They were pressing paper and then checking the scaling of the folding with the volume and then the number of papers that they were using. So again, the limit in space would force the fold to the brain to fold. Now there's some other type of mechanical hypothesis. This is like what I numbered here, hypothesis 2, where it's a paper that was also in Science, that the argument was basically that brain folding is not crumpling. So it was interesting to see that many decades later, the idea of crumpling was still very alive. And here what they propose, based on observations from cytoarchitecture of the cortex was that there was a difference in the expansion of the superior and the inferior layers of the cortex. So a difference between layers 1 to 3 and then 4, 5, and 6, right? And so because of this difference that they were able to measure, that would produce a backlinking instability that would be a main theme in what follows, that would lead to brain folding. However, like just by measuring the difference in extension of the superior and inferior layers in the cortex, it's not enough information to know what was the cause of that difference, because by necessity, if you fold anything, the perimeter in the outside has to be smaller than the perimeter in the inside. But what was interesting in that previous paper is that the idea of the folding being an intrinsic property of the cortex appeared very, very clearly. Now, another way of explaining this clear intrinsic process is what I have termed here the gyrogenetic hypothesis, which are very much some sort of bulging, So, well, or at least some part of the gyrogenetic hypothesis can be considered as bulging. Probably the first time the idea appeared, or at least that I am aware of, was in two papers by Smart and McSherry in 86, where they were looking at the development of the folding in ferrets. And very interesting, what they observed, that you can see clearly in the picture to the left, was that as soon as fold appears, the position of the sulci doesn't seem to move. And it's only the gyri that seem to grow outside. So that led them to think that probably the growth process is only localized within gyri by some patterning mechanism. The idea was taken to a really a much more developed place by Wally Welker in two papers, one that addresses the folding of the gyrogenesis in the cerebrum, and the other one in the cerebellum. These are so probably two of the most beautiful papers that I have ever had the opportunity to see, and I really encourage you to look at them and try to find them. So the idea is that the gyri, in a sense, are like organs in the cerebrum. They really divide the cortex in the cerebrum and the cerebellum in modules with very specific function, cytoarchitecture, and connectivity patterns. And the idea, based on the observations by Smart and McSherry, was that this was a genetic process. So in the same way that we grow arms or eyes, we also grow our gyri in a very species-specific pattern. Here you can see some of the pictures. Really, it's amazing papers to read, and you will be delighted with the beautiful images and illustrations. So there's been a couple of models or reformulations of the idea. For example, one could imagine that, and it has been proposed. So when you look, if you have ever looked at reaction diffusion patterns, they seem to look like in their geometry like brain folding pattern. So this idea was developed by Julien Lefebvre and Jean-François Mangin in a paper in 2009. And so they have this reaction diffusion mechanism, and then somehow the morphogene that's diffusing, depending on the value, will be associated with increasing in thickness of the cortex or not. Right, so it's in a sense a gyrogenetic mechanism, because it depends on this morphogene to lay down where folds will appear. But it's only based on the reaction diffusion of potentially only two morphogenes, right? But one could imagine that this could be the case with a different sort of mechanism, with more specific regionalization of the folding pattern. An idea that was proposed or described by Lisa Ronan and Paul Fletcher in this paper, and they call this the differential expansion or differential tangential expansion hypothesis, which is a bit unfortunate because it has led to a lot of confusion between the language that's used by physicists and the language that's used by neuroscientists. So here, what Ronan and Fletcher mean is that different parts of the cortex will have a differential expansion in a sense that's probably more similar to what Martin McSherry and Welker were thinking. So the different regions that would be genetically determined would be determined to grow more or less, and then that would lead place to folds, as you can see in the figure. I will go back to that to describe the meaning that physicists give to this differential expansion wording, right? It's very interesting that the team of Victor Borrell in Alicante was able to show some evidence of the existence actually of this sort of genetic heterogeneity that would, in a sense, precede the formation of folding. Here in a paper from 2014, they were looking at the ferret neocortex, and they observed that at P6, a time where it was difficult to see any folding, they were already able to see a heterogeneous transcription of some important genes for brain development. So the idea would be that this heterogeneous pattern appears first, and then it leads to the formation of folds through this bulging mechanism. However, that may not be completely the case. We have been looking at ferret brains, and probably as early as P2, which is 2 days after birth, we can already observe some changes that can be measured in MRI that seem to precede the formation of a folding pattern, although it's difficult to see with the naked eye in a brain slice. Okay, another very popular gyrogenetic hypothesis, especially in our community, is the axonal tension hypothesis proposed by David Van Essen in '97. So the idea was that different brain regions would be, as you can see in the figure to the left and the bottom, would be, I can use my mouse, no? Would be programmed to be connected. No? So there would be a connectome for the brain that's genetically encoded. If some regions have been programmed to be connected, and if their connection is strong, because of a process of minimization of force, those two regions would be forced to become closer together, turning into a sulcus. And what he observed is that regions that have been shown to have a very strong number of connections seem to be on one side and the other, one wall and the other of a gyrus, as for example here in V1. Some researchers have tried to formalize this idea, and this is the work of Klaus Hilgetag and Helen Barbas, who show that, but this is not really surprising. If you were to have connections, and if they were to shrink, that would lead to a fold. Okay. However, there's a lot of experimental evidence and also theoretical, but this is strong experimental evidence showing that axonal tension, it doesn't seem to be the trigger for brain folding. So first of all, as I was describing at the very beginning, the most frequent pattern that people observe is not a pattern that, as would be predicted by the axonal tension hypothesis, that would be like this one, but more the U fiber, which is like the complete orthogonal pattern to what would be expected. Plus, in this paper, the authors, which are extremely influential biomechanicists, were doing indentation experiments where they cut a piece of the cortex of a ferret or a mouse, and they observe, so if the cortex were pulled by the axons, if one cut through it, one would expect that it opens up, and what they observed was exactly the opposite pattern. So this is very solid direct evidence of the incorrect idea of that axons would pull on the brain. The title even is very clear. However, it's still unknown for many people, probably because not all neuroscientists read the Journal of Biomechanical Engineering. So another hypothesis that I number here, hypothesis 4, is one where we have been working for quite some time. It's very similar to the idea of the Richman model, where, if you remember, it was the superior and inferior layers of the cortex that would lead to folding. But here it's to recognize that actually the two different materials at play are the cortex and the white matter, right? Which is basically the place where all the bodies of the neurons is located and all their axons is located. So it's actually the same cells, right? But two different parts of the cell. So they are strongly sticking together because it's basically the same cell. The model is very simple, much more parsimonious. So it only requires growth of the cortical layer and elasticity in the cortex and the substrate, as you can see here. So this is the formulation as it was proposed in 2005. Then this is the same idea extended to 3D in 2012. And this is our most recent results. What we are using at the moment for modeling brain folding, where it's in 3D and we can regulate different parameters of the geometry, leading to this sort of simulations. But we can also change, instead of using an ellipsoid, and we can use an initial geometry that looks, for example, here like the initial geometry of a ferret brain. And then if we trigger our growth model there, we observe the creation of a folding pattern that in many aspects resembles the folding pattern of a real ferret. So I will describe this type of modeling more in detail because it seems to me that's the one that's closer to what we observe in real brains. So it's what I would call the biomechanical models of cortical folding. Okay, so for this, I will briefly introduce the basic ideas of elasticity and growth modeling in biomechanics. So first of all, you may remember from high school the Hooke's law for a linear spring. So the idea would be that the amount of force required to pull a spring is constant, is proportional to the amount of deformation that you introduce. So the stress would be proportional to the strain. And here in the illustration, you see the effect of a linear spring of adding one weight or two weights, then the deformation, so the strain would just double linearly, right? Now, if you are not using a linear spring anymore, but a piece of like a 2D piece of rubber or a 3D volume, things become a bit more tricky. In particular, because if you may have the experience of pulling a piece of rubber, it will not only stretch in one side, but it will shrink in the orthogonal direction, right? And that's what's called the Poisson ratio. So first of all, the how much you need to pull to get some amount of deformation would depend on Young's modulus of the material. So here I give some ideas. In aluminum, it's measured in pascals. Aluminum would be like 70 gigapascals. Bone, about 1 gigapascal. Then jelly, it's like 30 kilopascals. And cells and brain tissue are extremely soft, so even softer than jelly, right? So brain tissue can go from just 100 pascals to 10 kilopascals. Now the Poisson ratio will determine how much a tissue normally shrinks when you pull. That would be in the ideal case where the volume is exactly conserved, would be a Poisson ratio of 0.5, and brain seems to be like about that order. Now you have some other materials like cork, where even if you push or if you pull, they don't seem to get deformed in the orthogonal direction, which makes it very useful when you want to close a bottle of wine, for example. So the Poisson ratio for cork is 0. And there's even some weird materials, offsetting materials, where the Poisson ratio is negative. And here you have a little example just for fun. So if you imagine a material that's built like in the picture at the bottom, and if you pull to the sides, instead of shrinking, it will expand. So that's very funny. So now if you want to make a linear model for elasticity that includes both the stiffness but also the Poisson's ratio, then you end up with an equation like the general form of the Hooke's law for linear elasticity, where you have a stress that depends on strain, and then the mu and lambda are Lamé constants, which are related to the Young modulus and the Poisson's ratio. So that's the way in which you expand the linear elasticity from high school to linear elasticity in more general solids. Now, it's interesting also to think in elasticity in terms of elastic energy density, and not only in terms of the stress and the strain that you have to use. It's very interesting to decompose the deformations of whatever object you are dealing with into changes in shape and changes in size, because they will have different weights in terms of elastic energy. So in different materials, elastic energy W will be differently affected by changes in shape and changes in size. And then you would write the energy like this. And in mathematical terms, this can be written in this way, using F, which is the deformation tensor, and J, that's the determinant of the deformation tensor. So that determinant will indicate how much the local deformation is expanding or contracting your element of volume. Here you have some examples. So imagine that you start with a piece of a rectangle, and then you impose this deformation that you see at the bottom. What you see in wireframe is the resting state. So what the piece of volume would go back if you stop deforming it. And the deform configuration is the one drawn with a solid line. And you see different deformation tensors. So first, in C, you see one that's only increasing the volume without changing the shape. Then the second one is changing the shape through a skew without changing the volume. And then E, it's doing both at the same time. So that's to give you an intuition of what this tensor F is doing. Now for growth, and this is very interesting. So the way in which you can model growth is by modifying the size addressed of each element. So imagine that your element would be addressed at the beginning if it were like slightly larger. Now it has to be much larger to be addressed. So that will make the actual size of your object to grow. Right, and this leads to a very nice number of unexpected results. So in terms of tensors, using the same notation as before, it would be written down like that. So it's a multiplicative decomposition of the deformation into a part that would be due only to elasticity and another that would be only due to growth. Now, if you have configurations like the one that I was describing with a cortex in a substrate, this can potentially lead to a buckling instability that leads to the formation of faults. And so physicists describe different depending on the ratio of the stiffness of the substrate and the cortex. It can lead to different type of geometry. So if the layer on top is stiff and the tissue at the bottom is soft, that will lead to wrinkles that look sinusoidal. Now, if the tissue, the substrate is very, very soft, imagine like something growing on top of liquid, then it will lead to the formation of what physicists call a fold. That's a very, very deep fold, a very indentation that can progress deeper without being perturbed or impeded by anything. And if the stiffness of both the cortex and the substrate is more or less similar, then that leads to the formation of creases that some physicists call also sulci, but it's not what we would call sulci. I think that from experience by looking at brains, the type of instability that we're observing seems to be something more similar to wrinkles, right? And here you have images of different places where these different type of versions of the buckling instability appear. Imagine now that you have this piece of rubber that's like a disc, and you make only grow the outer rim. But now you stick them back together as if it were the neurons and the axons of our brain. That would lead to folding, because it would be the way of making compatible the growth of the outer rim and the elasticity of the core. And what's interesting that I'm showing here is that also leads to a gradient of a pattern of residual stress. So now your object, even if it's not growing anymore, it will not be addressed. Well, it will be addressed, but if you cut it, it will jump. Imagine when you have like a very ripe watermelon and you push your knife and then it breaks open, right? It's the same idea. So in this tissue there will be a stress that's never released. So for the modeling, the probably most classical idea or formulation where this sort of problem appears is a Winkler foundation in engineering. So it's the sort of model that people use to model a road where you have the top layer, that expands and sometimes wrinkles, and it's sticking to the substrate of the road, right? Um, a description of the relationship between a Winkler foundation and a model of brain folding was first described, as far as I know, by the team of Phil Bailey in 2013, that you can see here. In particular, if you fit to this system of a cortex, here it's not a circle, but just a block, and you're pushing from the sides, if you fit a sinusoidal solution, you would be able to come up with a formula that would predict the wavelength of your fold based on the thickness of your cortex. That's the one on the bottom right. So this is the most simple version of a brain folding model that I could imagine. So you have this cortex and a core. Now, if only the cortex is growing, the configuration without fold becomes unstable, and it leads to these brain folds with a wavelength that will depend on the thickness, following something that's similar to the formula that you saw in the previous slide. So it's surprising how such an extremely simple model can reproduce or give an interpretation to a lot of different observations in neuroanatomy and in the neuroanatomy of brain folding. So first of all, neuroanatomists have described different phases in the growth of a brain, where first brains will grow without folding, and then at some point folds will appear and then become deeper. And you can observe also the same type of phenomenon in this very simple brain folding model. So at the beginning, it will be mostly the brain growing without folding through compression. And then at some point, when compression becomes impossible, you will have wrinkles that appear. It's also interesting to observe that the bottom of the solsi appears not to move, which is probably what Smart and McSherry were observing, or could be an interpretation of their observation that's purely mechanical this time. Now, you can also observe that when you have poles that appear in this sort of configuration, solsi will become thinner than gyri, just because of the mechanics. No, this was observed in simulation in 2005, but also later on in polymer simulations. So where you put two different gels and then one gel is made to swell. You also observe that in all cases, solsi become thinner than gyri. You can also observe by making models of different sizes that now the relationship between the perimeter, what would be the surface, and the area, what would be the volume in a 3D brain, seems to follow allometric rules. So normally if brains were growing without changing in shape, the relationship between surface and volume would follow a scaling factor of 2/3. But what you observe is that actually the scaling factor is more or less linear. So the emergence of this linear scaling factor also appears in these very simple mechanical models, as you can see in the figure. You can also observe something that could provide an interpretation for the sequence and characteristics of primary, secondary, and tertiary folds. So if you have a brain fold and then you perturb the brain the brain model in one specific point, you will observe a fold that appears at the position of the perturbation, and then a cascade of smaller folds that grow to the side. And that seems to correspond to the observation of primary, secondary, and tertiary folds, which appear progressively later and are progressively less deep, right? Now, this could be in a completely symmetric model triggered by a perturbation, but it's also interesting that the geometry of the initial shape can determine the place where faults or the primary faults will most likely appear. So if instead of using a circle, you use an ellipsoid, then because of the geometry of the ellipsoid, without need for any additional perturbation, you will also trigger this cascade of primary, secondary, and tertiary faults. You can also observe this in 3D. And then here you see that faults seem to appear or align with the major axis of the ellipsoids. Later on in 2014, in a beautiful paper by the team of Mahadevan, Talinen showed that a similar effect. So depending on the shape of their models, the orientation of the faults would be regulated, would be aligned to the geometry, and that was later on demonstrated even more by using as initial shape for the brains something that looks like a very early fetal human brain. No, and in the simulations or in polymer models made to swell, you also observe this formation of faults that seem to align with the initial geometry of the model. So this could suggest that even the folding pattern, and not only the folding, could be somehow regulated by the geometry and could be purely, in a sense, purely mechanical. Okay, so that was basically what I had to show you. And now, if you want to go further, I would have some suggestions. So first, I would like to just make some questions. Probably now, based on what we have seen before, you could have some ideas. So what would you say? Why the brains of mice and rats are enfolded? I will not answer that. So why are the brains of manatees? You know, manatees are quite big, and the brain volume is about the size of the brain volume of a chimpanzee, but their brains are perfectly smooth. Why could that be? What could be the possible explanations? Now, you may have been able to see at the butcher probably the brain of a cow or a sheep, and they are extremely folded, like much more folded than the brains of carnivores, no, and the brains of dolphins even more. So why could that be? What could be possible explanations for their extreme amount of folding? And now, finally, and I think it's important to make this question, people have brains with varying degrees of folding. No? So some people, like most likely people with large brains, have very folded brains, and people with smaller brains, they will have like less folded brains because the allometric scaling rule is the same for everyone, be it human or different species. Does that mean something? Okay, and I will leave that for you to think. So now, if you want to investigate more about the biomechanics of folding or the problem of the different hypotheses of brain folding and the consequences that it may have for the organization of brain structure and function, I would suggest two reviews that we have done relatively recently. One with Ophélie Foubet, that's mechanical morphogenesis and development of neocortical organization, and another one that's more theoretical and then try to consider the impact that this may have or the role that this may play in the development and evolution of the neocortex. Now, the biomechanical models are one possible explanations, but if you want to review alternative models, then I would suggest that you look at the recent review by Victor Borrell and Linares Bernadero, deconstructing cortical folding, genetic, cellular, and mechanical determinants, where they will describe much more in detail all the genetic explanations for the formation of brain folds. Now, if you're interested in the more recent developments on the idea of axonal tension, then I could suggest that you read the recent review by David Van Essen, a 2020 view of tension-based cortical morphogenesis. Now, if you want to go further on the thinking, so the ideas behind all this research on brain folding, then I would, of course, suggest you to get a copy of On Growth and Form by Darcy Thompson. Then also this the idea of, you know, the debate between a genetic determination or a developmental constraint. I think it's very nicely introduced by this paper, famous paper by Gould and Lewontin, The Spandrels of San Marco in the Panglossian Paradigm, a critique of the adaptationist program. And going further in the type of thinking that in a sense tries to challenge a purely adaptationist or selectionist view for organismal evolution and development, then the book by Pigliucci and Müller, Evolution, the Extended Synthesis, may be interesting. And finally, if you're really, really interested in this subject, we are just starting together with a fantastic team of colleagues, an ERC Synergy grant, and we will be very soon hiring postdocs if you want to come work with us to work on the modeling of brain folding and also in the description of brain folding in ferrets, mice, frogs, chicks. You will have the opportunity to, well, you will be working with me, but the idea is that we'll be working in synergy together with the team of Victor Borrell, Laurent Nguyen in Belgium, and Christian Franz, who is a fantastic mechanobiologist working in Germany. So thank you very much.